Good evening, everyone. I am delighted to welcome to you all today insightful online session on building hypotheses from the traditional claims and literature data. I am Miss Sakshi Dabe. I will be your host for this evening. We are truly honored to have Dr. Sriharan sir with us, a distinguished expert researcher, and they will be a guide us through this session. Now I request to Dr. Sriharan sir to carry this session forwards. Thank you.
Hi all, good evening. Yesterday class we see about how to map the herbal trail on WHO and clinical trial.gov government database. So the yesterday class is based upon how to analyze how to map the herbal medicine, what are the quality assessment takes place. Likewise we see. So followed by the yesterday class, we are going to see about today how to build the hypothesis from the traditional claim and literature data. How to we build, so today class is going to see about these are the things. So followed by this class, we are going to see lot of things, learn about how to we execute our mapping. Likewise, we can learn more thing on today's session. So, followed by the yesterday class. So, scientific inquiry. So, scientific inquiry is nothing but our identifying there our data in the data in the literature database. We are identifying that. So, the scientific inquiry is very important. So, this give the progress of careful review of our evidence and traditional medicine. It will give the careful review on that. So today's session is very useful for you. So through the session we can see how to develop and how to transform our claim into scientific testable hypothesis. So today's session is fully based upon our traditional claim and literature data, how to transfer these into hypothesis data. So we are going to explore a lot about things today. So understanding the traditional claim. So the traditional claim is divided by these three category. The culture knowledge, it is a background of culture knowledge system and historical precedent and analytical evidence. So culture knowledge system, you may know about that. The herbal medicine used by over a year, over a century of the year. So it can give confidence to the people, and it also give confidence to do research on that. So due to the background of culture knowledge system and historical precedent is nothing but it give the data documentation of how, when and where, how their medicines are used, how they are you can, based upon this historical precedent, you can compare the report with our scientific study. So it historical precedents also one of the thing. And another thing is anecdotal evidence nothing but it is otherwise called as a non-clinical PRO, patient report outcome. So that is anecdotal evidence. So it is a personal testimony about how they get, how what are the effect on them during they taking the herbal medicine. Likewise, they are giving testimony. So the role of literature data, you know about the literature data. Here the literature data we are dividing into two types. One is primary source and secondary source. So the primary source are nothing but the data are taken from the original research publication article, taken from the clinical trial. So these are the primary source. So secondary sources like a review article, systematic analysis, meta-analysis, likewise case series, case report. These are the secondary source of literature data. So this is the main part we are going to see about that. How to construct the hypothesis framework, hypothesis construction. How do we want to do? So today's session is we are going to see about how to test the hypothesis. So testing hypothesis is main important. So how to build the hypothesis means hypothesis is very important. So first step is observation phase. So observation phase is nothing but we are gathering the data. We are gathering the data based upon the traditional medicine and literature review. We are gathering the data on that. That is the observation phase. After observation phase, we are move on to the pattern recognition. In pattern recognition, we are identifying the trends. In pattern recognition, you want to think main thing, two things. What means what are the successful outcome is repeated. What are the we mean the successful outcome repeated back can be used as in our research also. So in pattern recognition, you are analyzing the successful outcome repeated, and also you are analyzing the inconsistency. What are the inconsistency takes place during the research is otherwise called as a gap. So that is a pattern recognition. Is a second step. So after pattern recognition, the third step is hypothesis formulation. We are now going to formulate the hypothesis. So hypothesis formulation is nothing but we are converting the qualitative data into quantitative data. So here the qualitative data is what? Our database, traditional claims and literature review evidence is a qualitative database. So we are converting here the qualitative data into quantitative data. So quantitative data is nothing but we are giving a testable statement on hypothesis. We are giving a testable statement on hypothesis. What is our hypothesis testing? We are converting. So while your hypothesis should be based upon, it should be in a clarity and clear manner with build of scientific evaluation is very important. So this is the hypothesis construction framework. So I'm saying one example. Just imagine that. What is the example means? If you are doing any research on turmeric related field and that is used for inflammation. Likewise, your research is based upon turmeric and inflammation. Means what means? Here your observation base, your what going to be do means we want to search the data and gather the data related to the turmeric, which should be Ayurvedic text or should be likewise before I'm saying, it's a cultural background space or otherwise called as a testimony. You want to search that. So you want to search the data properly. After searching the pattern recognition. So the pattern recognition is the next step. So the pattern recognition based upon your research, systematic literature review. So the systematic literature review, I'm saying in a example for turmeric. So in systematic literature review, they have so multiple study. So it give the multiple study as a single result on that gathering of multiple study. So we can understand active component, supplement, how the inflammation acts and increase or decrease. This is the pattern recognition. So then your hypothesis is changing the qualitative into quantitative. So qualitative into quantitative. What this means? Your hypothesis questions based upon this example. What I'm saying is turmeric inflammation. You imagine this turmeric. We want to take daily as a turmeric. So daily standardization supplement. For that while combined with our another own research question is inflammation. So our daily supplement will of turmeric will reduce the inflammation. So this is our hypothesis with chronic inflammation biomarker. So biomarker. So main thing is our research question is turmeric and inflammation. Here, what is hypothesis? It means biomarker. So the biomarker is a hypothesis. Will the daily supplement will reduce the inflammation by what? Through the biomarker concentration alone. So here the hypothesis is biomarker. So followed by the next type. So type of traditional claim. So therapeutic claim is focused on pharmacology claim. So on another agriculture claim is focused on agronomic characteristic of the trial and crop productivity and any resistance to the pest and how the irrigation method should be improved through this herbal trial. Likewise, that is agriculture background. But our background is therapeutic climb is very important. We are for only focus mainly focus on pharmacology. So another thing is environmental observation. So environmental observations are most of the trials are conducted by ecologist scientists. So they all know about the environment. The studies based upon observation, environmental changes, species behavior and animal behavior, and what are the ecological behavior based on that we can see that. So this is a type of traditional crime. So followed by traditional therapeutic, agriculture, and environmental observation. So evaluating the source credibility. So this is very important. So based upon this credibility alone, we can access this. So first of all, you want to prioritize the source. Why means not all the sources are important. So not all the sources are want to important include in our study. So building the hypothesis framework. So source credibility is very important. Source credibility is very important. So our source credibility from the culture census, how the peoples are delivering the, from the decay, how they work, what are the benefit on that through their century. Likewise, we are cultural census and documentation quality. So what are the good documentation quality? Based upon the documentation quality, we can identify the standardization, how to want to follow the procedure method. We can easily identify the document quality and historical consistency, how it consistently through the long decay, how it consistently working. So this is evaluating the source creditability. Then literature review method. So literature review method is one of the important thing. Why means the effective literature is very important. Why means if you do the effective literature review means it will capture the all the relevant scientific evidence to you. So literature review methodology. First of all, scientific researching. You are searching from the multiple database like with PubMed. In clinical evidence, we go with PubMed. So based upon herbal medicine or a nutraceutical product means we go with the ethnobotanical database, likewise herbal medicine database or anthropology database. Here we are using anthropology database. While we want to get the historical culture content means we want to go to the anthropological database alone. Then alone we know how the past history, ancient history culture use the herbal medicine for their for their lifestyle. Likewise we can know that. And proper screening. So proper screening is very important, like keyword, like based upon and or not. Likewise it's a keyword Boolean method logic. Likewise we are good doing. And gray literature. Here why we are searching the gray literature means, yes, some of the gray or quality literature has been published in a paper presentation, conference presentation, which also give the weightage to the study. So that is a gray literature inclusion. So these are the scientific literature methodology. So critical observation is nothing but yesterday itself we see about the quality assessment, how the bias identification, how to overcome that mitigation, sample size and power, how to analyze that, and how the article is reproduced by following research. That is reproducibility evaluation. So identifying the research gap. So we know about that common research gap arise in the herbal medicine, sir. That is a magnetic understanding. We have a lack of understanding about the mechanism or action of the herbal medicine. So that is a magnetic understanding. And dosage response relationship. We cannot know about the pharmacodynamic, bioequivalence of the herbal medicine, when to use, how long to use, what are the discontinue, when to start, when to discontinue, what are the relapse store, we cannot know. And population variability. So there should be low patient requirement, low sample size. So that is a population variability. So bridging the world. So here we are going to see about how to what are the hypothesis characteristics. Here today's session we are going to test the hypothesis. So we want to know about what his characteristic. So hypothesis characteristics are three things: testability, falsibility, and parsimony. So these are the things are main thing. So testability, what it means, you want to simple, but you want to understand assurance or ensure. You want to ensure whether the literature data relevant traditional claims are came from the proper traditional claim. You want to understand why means here the testability is depend upon qualitative. In previous slide itself I'm say we are in hypothesis, we are transforming the qualitative into quantitative. So it analyzes the ensure the qualitative data. And falsibility is what means, uh, here the falsibility is very important. It means in testability, in language, in the word itself, it's something testing. So falsibility is based upon this falsibility, we can eliminate the incorrect idea. So our ideas may be incorrect. It should be eliminated by using this falsibility. So I'm giving example what means. For example, lungs cancer, smoking cause lung cancer. So you want to image that as an example. So if it should be proven by someone means it come under the falsibility. It should be, I mean, the smoking cause cancer. We are saying that as a statement. So someone is saying smoking also not part of the cancer. Smoking also is not a causative agent. It also cause another thing likewise. So we are disprove it or tested it. That is a falsibility. So they, you want to simply understand what means. We are proving the wrongly observation data, whether we are going to prove the some wrong observation data. That is a falsibility. So parsimony is nothing but it's saying about our hypothesis characteristic should be based upon simple and straightforward. Why means it is very important. Why means it is very important. Your characteristic should be based upon simple and straightforward. Why? For example, I'm saying uh, you are testing any from the example from the turmeric itself. You are compound. You are testing a turmeric plant to reduce the inflammation by blocking enzyme A. So this say the simple and straightforward hypothesis. This is a great example for parsimony. But in some cases, the parsimony is what mean we are using turmeric plant which reduce the inflammation. They are the several unknown way. Likewise, our component, the plant working, we have multiple unknown pathway. Likewise, we are collapsing and confusing. So this will cause the parsimony. So this is the parsimony. So from the observation to hypothesis, this is a step-by-step journey. So first of all is a traditional claim. This is an example, great example for that. Traditional claim, willow bark will relieve the pain and reduce the fever. As a traditionally, it called as a traditional claim. Based upon the literature evidence, in the bioactive compound, biological compound, the salicylate compound identifying as an anti-inflammatory property based upon the literature evidence. So, based upon the literature evidence, we develop the aspirin medicine now. So, this is the testable hypothesis. So, this is the great example of from the observation to hypothesis. So this is a case study of turmeric and inflammation. So traditional claim, it is Ayurvedic medicine used for the century to treat the inflammation condition and promoting healing. So literature, what literature support mean? Here the curcumin is active compound. It act as an anti-inflammatory property. In vitro study, it will give the data on that. So that is a literature support. So developing the turmeric hypothesis, what we are developing. So our research question is first important. So our research question is the curcumin or turmeric reduced the inflammation. Here the hypothesis formulations what means the daily curcumin supplement will significantly reduce the interleukin R and TNF alpha factor. Here, this is the biomarker is a hypothesis formulation. So our experimental design is a randomized control trial. Likewise, follow up. Likewise, this is the experimental trial design. So variable and operationalism. So this is very important. So you want to tell knowledge about that. So independent variable in this study, the curcumin dosage is an independent variable. So independent variable is basically what we want to know. Then alone we know what is independent variable in this study. So I am saying independent variable is nothing but we are saying our placebo versus experimental drug, it give any differences, that is the independent variable. So if any differences arise, that is independent variable. So we are saying that, in another word, we are saying what are the potential bias should be caused, should be controlled by the researcher, also independent variable. So that is also. So dependent variable is nothing but it, if we are going to measure or it show how our intervention work or not. So here our intervention work or not is we are going to see about the serum inflammatory biomarker interleukin 6, TNF factor and C reactive protein level. We are going to see about this biomarker whether this intervention work or not. That is the dependent variable. So control variable is nothing but other factor like that maybe influence that this dependent variable. Dependent variable here is serum inflammatory biomarker. So this control variable influence the dependent variable, that is a control variable. So in other word, what we say means the arrays of confounding factor will be happen here. So we want to control that based upon safeguarding that. So that is a control variable. So this is a variable and professionalism. So ethical consideration in simple we know we want to get ethical during our study research we want to follow that, we want to give the informed consent form like that, that is the ethical consideration. So here the cultural sensitivity framework are while we doing the research we want to do the informed consent form to the consent person, which should be in clear communication about research purpose and outcome and benefit sharing. Here benefit sharing while we doing a traditional medicine means we want to give the benefit sharing to the certain community or certain local regional to doing the research on that. So that is benefit sharing, cultural production. So some may mistrust ourselves, and some people are fear on us. So we want to overcome that, and we also want you the production to the cultural to production to the those people. We do not do any vulnerable activity against them, any rude activity against them, or forcing them to do into the trail. Likewise, that is a cultural production we want to ensure. And capacity building, give the training and education to our community collab members, and community engagement, collab partnership with the regional center, international policy. Likewise, that is a cultural sensitivity framework. So what are the common pitfall means confirmation bias. So confirmation bias is basically arrays that is otherwise called as a favorable result we are publishing. So we are avoiding the contradictory data we are avoiding. That is a confirmation bias. What is in our mind, what is in our belief, we are going to publish that. That is a confirmation bias. And cultural misinterpretation, mistrust. Some people are not trusted. Some people are have fear on us. That is also one of the pitfall. And a reductionism is what means in traditional system we know traditional medicine itself, we know that it should be a whole system. It is not to be in a one system or one small piece of system. So if you take the one herbal medicine means it can work as a three factor. It can work as lifestyle modification. It's also help mind, body. Likewise, it will help. So while we doing a research on one certain thing in one small thing is a reductionism in research on that. It also give gain while we doing a research on the one piece of the system. Also it will give the personal, it will also develop the personalized medicine to the certain herbal plant. So this is the quality assessment criteria. How want to, if you're doing any means the source reliability, how the cultural authenticity, how the testability. This is the graph showing that. So mixed method approach. So mixed method approach is nothing but combination of both qualitative and quantitative method. So here the traditional medicine or herbal plant have rich culture, rich essential sources on that, but their number and trails are very low, very low. So we are mixed method approaches. We are combining both. So we know about quality methods are ethnographic studies, participation observation, deep interview, focus on group discussion. We may know about what are the method we are going to say. But what is mean by the qualitative method means it will give understand why and who peoples are used. Why are we are using? Who are the peoples are benefit from this and how they are using. That we can identify using qualitative method. In quantitative method, is nothing but how well it work by measuring some outcome, some measuring the group. Likewise, we are going to see about that. So here the qualitative methods are controlled clinical trial, laboratory analysis, statistical analysis. These are the controlled clinical trials. So based upon this combination, it will give the good approach in herbal medicine. So literature landscape is we know database selection strategy, Medical database, we know the database are PubMed, Cochrane Library, MPACE. These are the medicinal data, medical database. In ethnobotanical source are based upon the herbal, pharmacognosy, species identification, botanical geology. Likewise, it come cross culture databases to be based upon the both ethnographic and anthropology. Here, why the both same means it comes under the one cross culture database. Why in anthropology is a ancient history. They're using some herbal medicine for their research. So here itself, the anthropographic also came here. How the geographical population highly which herbal medicines are used in ancient period and how they are used. How what are the medicine they are used and benefit through this herbal medicine, we can see on the cross culture database. So search strategy development, we want to develop the keyword. So using Boolean logic and or not. And what are the first main thing is you want to maintain the citation track and effectively. Why means while we miss the any citation means it will give a drawback to your study, and you also go back and search the citation, and it will also give the time lot of time also take. So while you seeing the any say citation, or you are doing any research article, review article, you maintain the citation tracking or citation method. So you that app may be mentally. So mentally is a app on which we maintain the citation. Otherwise we can maintain in a simple document itself, Google document itself we can maintain, so we can easily track that. So evidence synthesis technique. So this is the technique. First of all, we want to data extraction. We are taking the data extraction from the medical database or ethnometrical database, and quality appraisal means we are reviewing that, peer reviewing and analyzing the study design, sample size. And third thing is pattern analysis, outcome measure, whether the successful, what are the repeated successful outcome, what are the inconsistency. We are doing that. So that is a evidence synthesis technique. So meta-analysis, you know about that. It is a combine of multiple study. So it can give the strongest conclusion on the herbal medicine. So here the study heterogeneity we are accessing using the meta-analysis in about any variation, patient demographic, intervention, outcome measure. So we are accessing through that. So it also give the power packed result also. Statistical method, it's one of the important thing. Or what method we are going to select. You based upon this meta analysis, we can get idea. So the lot of study combined in a one single result or one strongest result, so we can easily get through the statistical method. And publication bias, so what are the publication bias, what it help to adjust the bias based upon this meta analysis, we can adjust the bias. So this is the meta analysis consideration. So this is a case study, how the case study ginseng research evaluation takes from traditional use to the clinical trial. This is the timeline. So the traditional use, you know, it about over the 2000 years is we have been using for vitality and cognitive enhancement. So the early research in 1950 is based upon the stress response modulation and up to genetic property, you know about that cognitive to enhance that in 1950 use. So mechanism study is very important. So the mechanism studies that takes place identifying the genocide active compound. Why means which is a major active compound which will use for which affect the neurotransmitter system. So in 1990, research takes place on that. In clinical trial, the 2000 have a systemic study examining about the cognitive performance, fatigue, immune functions that affect research evolution takes place in genetics. So here the ginseng hypothesis development, how the hypothesis development from the traditional claim to modern hypothesis. Our traditional claim, here the timeline itself, you know about the cognitive. Here the mental clarity have that. It have a vitality in health individual. Likewise, we can in traditional claim itself, it is a mental clarity. It enhances the mental clarity. We know. So how we are developing here the modern hypothesis is, so our standardizing extracting these extraction, it will help to improve the cognitive performance. Also, it reduces the fatigue in controlled clinical setting. While doing control trial, it will deliver the good effect on those. So this is the modern hypothesis example on that. So the research design implication, how to implicate that. So the population selection is first thing and major thing. How define the inclusion population, exclusion population. That's the main thing on that intervention study. So it should be in a standardized protocol to follow the intervention study, how to develop, what are the extraction, preparation, dosing protocols are the majorly in the intervention design. So outcome measures would be we have a validating tool or validating instrument is very main thing. So yesterday class episode we know we do not use the multiple instrument for the one study or one disease. Example, the pain, we having pain, use one scale is one instrument is very useful. So appropriate validating instrument selection is very important. For here the cognitive impairment we are selecting, and duration planning, appropriate treatment plan to follow period is very important for the cognitive impairment patient. We have a long follow up if we want to need that. So research integration. So here the paradigmatic difference what means in traditional knowledge it should be based upon whole system, but in scientific method it should be a reductional approach in one piece system alone. So context dependent application nothing but individual effect. Some may get effect through this herbal medicine, some may not work. So the traditional knowledge based for context in the independent application. So in scientific methodology we have a standardized protocol required, then only we can move to the clinical trial or research or anything we can move. So standardized is very much important. And traditional knowledge have experimental validation method, real life experience. So here we want in statistical method we want a statistical validation method. Likewise before slide is we know selecting validation instrument likewise. So individual variation acceptance it will be likewise before the context independent application likewise individual variation acceptance. So the population generality is very important. Then only we can see about the broad about nature on that. So bridging methodological gap. So what the main gap is systemic biological approach. So developing omic data about proteomic, genomic species on that. Likewise, we want to that is a approach when we developing that we can identify the patterns of species in very much manner on personalized medicine. So likewise before class itself and the pharmacogenomic genetic variation should be takes place from the geographical location. So that is a personalized medicine. So mixed method approach, it will give the power pack result on methodology. So these are the bridging methodological gap. So hypothesis refining process. So first of all, we initially formulating that we are getting the broad hypothesis data from the literature or literature review and traditional claim, and after that we are peer reviewing that through the export evaluation. We are peer reviewing that. So after peer reviewing, we are start as a pilot and expanding as a final hypothesis. So pilot studies or small study. Based upon the small study, we are going to have access the feasibility, how the methodological design and how the adjust hypothesis parameter. Likewise, we are going to look on that. And after we are conclude these session, we are moved to the final hypothesis. After the refine of the pilot testing, we are moving on to this. So to the full-fledged, full-scale investigation. So this is the method from the initial formulation to final hypothesis. So this is the statistical power, how they are getting through this, the range of statistical, how difference on that. So technological integration. So technological integration here, texting using AI, we can understand the unstructured traditional document. Likewise, we can identify and analyze using this AI. On molecular modeling is very important to identify to understand the magnetic nature of the study, nature of the species. Likewise, we can understand the bioactive compound, what are the interaction purpose. Likewise, so data integration here, what means data sharing? Why means it will very helpful to data sharing. So that is the technology integration. So collaboration of research model through this academic institution, indigenous community, government funding and agency, pharmaceutical industry company, and non-government organization (NGO). So based upon this collaboration, we can get a research model in an effective manner, and successful hypothesis also we can develop on that. So this is the validation in strategy. So in magnetic strategy, what is your strategy validation here? You understanding biological pathway, how their biological pathway we want to understand. Clinical study based upon animal model and in vitro study. So clinical trial strategy, phase one, phase two, phase three is come under three, and population study and the implementation search implementation research. So this is the validation strategy followed by the hierarchical one, two, three, four, five. So publication and dissemination. So in academic outlet we have a low impact or low journal which should be published the herbal medicine or a nutraceutical product in their medical journal. So in academic based upon this collaboration or input, we can give the more peer review journal can be give more specialization in ethnopharmacology, traditional medicine. Likewise it will give way to this, and community engagement also sharing based upon their research community, regional community alone. They have a full knowledge about the herbal medicine. Based upon their engagement will give the new initiative, new idea on that. So this is a publication and dissemination. So this is the impact assessment framework. So there should be 85% have validate assessment, and it has a successful methodological rate. And 12 year here the 12 years is a average hypothesis development. So how the average hypothesis development in a clinical setting, it will take 12 years. So average research investment is 2.1 million. So this is the impact of the research framework. So our future direction. So emerging the technology using machine learning, we can identify the identify the trends based upon this machine learning. We can predict the research on that. And genomic integration is very important. Omic data. Based upon this integration, we can easily identify the mechanistic studies of the herbal plant, and the digital documentation using virtuality. Likewise decentralized style into likewise DCT implementation on that. So regulatory consideration, so research ethic, so institutional review board for herbal medicine should be happen, and yeah, they have involved those who are in research ethical committee. So those who have should be there should be a pharmacognosist or well known about background of pharmacognosy. Likewise, the intellectual property is very important. Why means based upon this alone, the most of the pharma companies are not much interested in doing research on that. So return on investment is very low. So protecting that them and clear rules on intellectual property will gain more research on that. And international guidelines about CBD, Nagoya Protocol, other international framework, which will give strengthen to the regulatory. So these are the regulatory considerations. So success matrix graph this happened how which the study more majorly taking place where the success matrix belong from the following pie chart we can easily visible on that. So common challenges as a solution. Language barriers is first thing. OI means herbal medicines are developed from the regional based alone. So language barriers also main thing. So using natural language browsers or using a lot of initiation has been take place with translation. We can overcome that. So cultural mistrust. So building a stressful relationship, we can overcome this cultural mistrust and funding limitation. So based upon this intellectual property and funding mechanism through the collaboration, we can also overcome this funding limitation. So training and capacity building. So giving adequate training to the concerned staff and research coordinator, clinical research coordinator, researcher. Likewise, it will give gain to them how the methods are used and standardization. And community partnership, collabing with the local center and regional center, and align with the natural our health policy. We can get more funding on that based upon this community partnership. So mentorship network, connecting a research scientist from the recognize organization to mentor their study. Likewise, this is mentor network, mentorship. These are the things will happen in developing capacity building in the training. So quality control measurement. The first thing is standardization. So the essential standardization protocol should be prepared for the traditional medicine analysis, and authenticity is nothing but we are verifying their material through the correct resources. Why means the source variability also happen will be raw material will taken from the herbal medicine. So authentication and documentation whether this your hypothesis should be reproducible, ensure that. So these are the quality control measure on that. So global research network established, the International Society of Ethnopharmacology. So it will give the based upon this establishment, WHO and natural product network. Based upon this established network, we can share the resource in a collab manner. Data sharing also takes place, identifying the standardized methodology and knowledge exchange platform also through this established network platform we can gain that. So innovation in hypothesis testing. So based upon the innovation of this hypothesis, so the adaptive trial design is very important and initiative thing in the hypothesis testing. Why the adaptive trial design I'm saying means it is a flexible trial. We can alter, we can adjust the study based upon our modification. We can adjust the study based upon our intent result, and we can also modify our hypothesis. No, in adaptive trial design we can start we cannot start from the first. We can go with whether the area we are stuck, we can move on that area alone, on from the area alone. So biomarker development, we can identify the novel identity biomarker. What means biomarkers main thing. So based upon this hypothesis, biomarker identify and real world evidence to adapting the clinical integration on clinical setting. So this is the innovation of hypothesis testing. So risk management strategy. So first thing is safety monitoring. So safety monitoring, we have adverse event tracking is very important to analyze the risk on that. So quality assurance, so standardized preparation protocol to attain to quality assurance, and the ethical oversight is we want update frequency. Likewise, before glad itself, we see about quality assessment framework that is takes place in ethical oversight. We are update the frequency continuously updating to the concerned ethical committee. So regulatory complaints align with the local regional regulatory community, local regulatory community. Likewise, we are other with their regulatory complaints. So knowledge translation. So here the knowledge translation is nothing but implementation of policy development. So about herbal medicine, adapting the national policy or regional policy. Likewise, informing the policy related to it, and clinical integration. So it will give the way to the evidence-based traditional practice on the modern healthcare. So clinical integration is also implement pathway for our future and community application. So returning the research from the community application from their community itself accessible format, we are getting the research on that. So yeah, this is the implement pathway. So future research priorities are personalized approach. We know about the personalized medicine develop based upon one piece system we can develop on that, and combination therapy. So in previous class itself, we say about the supportive care, and it also pay way to the combination therapy. Why means in one single one in herbal product have multiple ingredient. So based upon their ingredient, we can develop the combination therapy on that, and prevention focus about their lifestyle modification and chronic disease. Based upon the health policy, we are making the prevention focus on that. So this is the future priority. So see, this is the picture of the synthesis and integration of the omic of the hypothesis. So today takeaway is, so the respectable collaboration is very need from the community, regional center, and health stakeholders. So very important. And methodological awareness also we can learn, and how we want to systematically approach framework and mapping in next day class itself we learn about that. So today's session is based upon how the hypothesis should be developed from the literature search and traditional claim. Based upon this, we are promoting, we are develop how we are the steps are follow, what are the quality assessment takes place, or the class today we see. So today class is about on that. So you can you yesterday while combining with both yesterday class and today class, you can figure out the something about the herbal medicine, how to map that, how to analyze that, you can easily figure out that. And another one thing is hypothesis development. So in this slide today's session, we also look on to the hypothesis, how to develop the hypothesis, and what are the features and what are the quality assessment should be takes place. So these are the things when we doing a hypothesis testing, these are the things we want to follow that. So thank you.
Thank you, sir, for the sharing their valuable, insightful on the evidence-binding hypothesis from the traditional claims and literature data. Your expertise has been truly enriched this session. Sir, few questions are asked their audience.
Okay, ma'am.
How can the traditional medicine claim be scientifically validated using the evidence gathered from the existing literature data?
Yes, ma'am. So the traditional medicine, here the question is how they are scientifically validated, that like they are asking. So in the literature database itself, we can get the more answer on that. So a systematic evidence based approach alone that should be published in literature database. So we can get the data through the existing literature data, which will also use as a uh, uh, claiming purpose to undergo the ongoing research studies also. So in literature database, we can get the systematic evidence-based approach on that. So for example, the ethnobacterial record, which should be also used as a healthcare claim, traditional claim we can also use, and traditional medicine contacts like Ayurvedic, Siddha. Likewise, these are the records has been published in literature database. So we can get data which should be scientifically evaluated on that through the database like PubMed, Scopus, like that.
Okay, sir. How is important is statistical or bioinformative support in the validating hypothesis from the literature data?
How is important to statistical and bioinformative support in validating hypothesis from the literature data? Yeah. So, so in our literature data, we are first of all we are observing that, and after observing, we are observing our data, gathering that. So after gathering that, we are second step itself, we are changing that into qualitative into quantitative. So the transformation of qualitative into quantitative is very important. So based upon this meta-analysis itself, we can identify the statistical power. What are the statistical method they are used, how they are overcome that, and how what are the implement they are takes place. Likewise, we can use that. And here the statistical support is what means we can also see the significant testing also, true effect and false effect also we can test. So and based upon this statistical power also, we can identify the bias and what should be controlled, confounding factor also we can identify that. So the role of this bioinformating also very important thing.
Okay, sir. Thank you all once again. I and have a great evening ahead. Thank you so much.
And thank you, ma'am. Thank you for the opportunity.